Immunoprecise Antibodies is the full name of the company. It is a multinational company that exists really to solve the major demands that exist in pharmaceutical and biotech companies with regard to building novel therapeutics.
Hello there. Welcome back to the My Future Business Show. I hope you're doing wonderfully well today. It is always a pleasure to have you with us on the show. And if in fact this is your first time joining us, hello, welcome. I'm sure that you're in for a treat today because on today's call, I have the pleasure of welcoming IPA CEO Dr. Jennifer Bath to the show. Welcome to the show, Doctor.
Thank you so much.
Oh, look, what a pleasure to have you here. Now, you and I are going to be talking about the company IPA, what it does, its core mission, and what the future holds for antibody development. And we're going to explore the value that IPA brings to the marketplace. So there's certainly, to say the least, a lot to unpack here. So first things first, where are you calling in from?
Well, right now I'm calling in from New York because I'm here for a conference that is tomorrow. And then here for the rest of the week just for meetings. But this is not where I live. I travel here for the week.
Fantastic. So where is home?
Home is actually in Fargo, North Dakota.
Oh, beautiful. So what is it that you love about the place? Is there something you could share with us about it?
About North Dakota?
Yes, please.
Oh, that's a great question. I'm not originally from there. I moved there right after university, ended up doing my doctorate in North Dakota. I was definitely the newbie to the state where you definitely have generations of families that live there. And it took me some time to actually figure out what I did love about it. I did leave there after 25 years raising kids, having an executive position at Aldebaran, which is now part of Danaher, and then moving on to IPA, I left and went to Minneapolis, and then I went to Des Moines, and I ended up going back to Fargo, which I wouldn't have expected. And now I can say that I really get it. I really appreciate, if more than anything, the people, how nice they are, the work ethic that they have. It's just a very, very tight-knit community that I don't any longer feel an outsider of.
Yes.
For a long time.
Oh, absolutely. Well, that's fabulous. And I love to talk about these things, Jennifer, if you don't mind me calling you Jennifer. What I notice is that, we've got unique individuals running organizations, but by and large, businesses are businesses and they don't generally change. But what I have found over the years of running the show is that certainly the people behind the businesses, the likes of yourself, are always unique. And that's why I love to explore this just ever so slightly. So thank you so very much for sharing. Now, in terms of downtime, it seems to me that you would be fairly busy. But if you get some downtime, what do you like to do? Do you have any hobbies? Do you follow sports? What do you think?
That's a great question. I don't really get much downtime. If you had asked me, 10 years ago, I would have been coming off of, really a 35-year stint of spending my downtime playing soccer.
Oh.
But about 10 years ago, I blew one of my knees out in an indoor game, which is fine. Maybe it was time. I went on to referee on the weekends professionally for a while at the collegiate level.
Yep.
And now I would say, boy, when I get free time, I have one child who is playing NCAA basketball. That can be fun if I have the time to attend the game. And I really enjoy. And then apart from that, this sounds a little bit silly, but I love reading like technical papers.
Oh, that's not silly at all. Absolutely. No, there are certain people in this world that is their thing. Absolutely.
That's enjoyable.
Tell me about going back to soccer just briefly. Were you an attacker, defender? What position did you enjoy playing?
I was a striker. Always a striker.
Taking those shots, I tell you what, that is a game and a half. If you can get it right, I tell you what, there's not much standing around, is there?
No, not at all. Not at all.
And given how fit you must have been to play at those sort of levels, what do you do over daytime? Do you have a routine to stay fit?
I never really had to think about that until, a couple of years after I wasn't any longer to play or referee. And then I really had to actively consider how to do that. And what I found was I found it really hard to motivate myself to stay fit if I wasn't in a competitive environment.
Yep.
So for me, that was really difficult. If I had a ball and a goal, there was nothing that was going to stop me But apart from that, I found it really difficult. So what I really ended up gravitating toward was, I mentioned that I love, reading scientific papers. I love working.
Yes, I
I
can
do.
understand that. Yep.
Yeah. So one thing that I ended up doing is, I had slowed down, especially in the cold weather, with any sort of like cardio activity. So my husband had noted that and then just said, I know like running and those sorts of things in those types of environments was really fun for you. What do you think about a treadmill where you could still like have your work or take phone calls? And that was something that had been promoted at my previous role. There was access to multiple treadmills with places where you can put your laptop or you can put documents. And people used it. And I thought, that's a great idea. So now
There
I
you
do
go.
that at home, and
I
I
love
just
it.
have 1 or 2 calls a day that are more casual internal calls where I know that's going to be possible. And
There you
yeah.
go. Fantastic feedback. Thank you so much. Now, when we talked about soccer, I think about I'm still playing soccer out the back with my kids. Now, going back to your childhood memories, where did it all start? What was your childhood like? And can you share us a positive memory about that time?
Oh, yeah. Well, where it all started with regard to soccer was my whole family was in soccer. My dad was coach, he was a referee. It was obligatory for every kid. I had 3 girls in the family all to play. So back in the day, we grew up in California, so it was common,
Yep,
right?
yep.
That most people did play early on. So you'd be 3 out there on the field with, a t-shirt that went down to your calves. We all did. And he coached us for a very long time until we were, a little bit higher in the club ball level. And so that's where it started. It was just kind of a family love for soccer. My parents played on a recreational team. My dad was very big into being educated about the sport that you're playing so that you know the rules on both sides. That makes
Yeah.
you a better player also, and
Mhm.
from his perspective, which I think is true.
Yep.
Yeah, so that's where it started,
That's
just really like a family activity. We loved going to tournaments together on the weekends and yeah, just enjoying the sport together.
Thank you so very much. Now you talked about inspirational people, parents, and the likes. Now, when we are growing up in those early formative years, we're obviously exposed to people that inspire us. Who was in your life that you can refer to there?
Oh, that's a great question. I don't mean it to sound cliche, but I would also still say my parents.
Hmm.
They had very different personalities, and I always said I was, kind of technically the youngest. We were 3 kids, but I also have an identical twin. So we were kind of born at the same time. We
Yeah.
always talk about who's more like which parent, and we always said I was probably the most like both parents out of the 3 of us, and I really felt that and saw that growing up, like different things from both of my parents that I very much related to and very much respected, and so yeah, I looked up to them both. I remember as a kid thinking into a ridiculously older age, I should have known better that my parents could do no wrong.
Oh, yours too. I thought that was only my parents.
So honestly, I really think that's really my truthful answer is
Yes, great.
that they
Thank you
just
so very much again for sharing. Now, as we shift gears a little bit, I think it's important for context, Jennifer, if you could share a little bit about, I guess, your educational and professional background with the
yeah,
audience.
absolutely. So for my education, I started at the University of Kansas. I majored in biology and I had an emphasis on human genetics. Um, and I really enjoyed that I had chosen the University of Kansas at the time because I was interested in gene splicing. It was just getting to the point where we understood really how to, PCR genes, so we could sequence genetic information, and gene therapy and DNA vaccinations were just starting to take a foothold. And so I was very intrigued by that. And as I went through the application process and narrowed that down, actually with my parents, we found that there were only 7 schools really that could offer that type of an experience, not necessarily in the classroom, but through a research opportunity. And the University of Kansas was one of them. So I had already a connection to the University of Kansas because my older sister had chosen to go there. At the time, I was going to high school in a Chicago suburb. Round trip flights were $39 round trip to Kansas City. So I had already been back and forth multiple times watching NCAA basketball games and championships at KU. So I had a connection. I loved it. And I knew that they could offer what I was looking for from a research and educational perspective. So that's where I started. And then I met who is now my husband, who was in graduate school at the University of Kansas. So fast forward 4.5 years later, he was preparing to finish his Ph.D. And in politics. And I was finishing up my undergraduate education. So I received a proposal, which I accepted, and went to Fargo, North Dakota, which if you had asked me before that, I would have struggled to tell you where it was on the map. I had just seen that movie that had come out. But, followed him, right?
Yes.
Knowing that, for me, that I, was committing to that and to him, and happily, and made that trip up to Fargo, North Dakota, and immediately started kind of repositioning what that graduate program was going to look like, because the schools I had applied to were all in the South.
And you weren't.
Yeah, and it turned out to be a great thing because I found a great program up at the North Dakota State University, which I wasn't familiar with previously, but given the funding record, the publication record, and the topics, I became really intrigued. And I also the opportunity to be in a cross-disciplinary PhD program, which was very appealing to me, combining 7 different departments across genetics, biochemistry, immunology. Took up a position in the immunology lab under the guidance of a gentleman named Dr. Michael Robinson, and did my PhD program there in cellular and molecular biology, and loved every second of that. And ended up doing my main research program on what's called chronic helminth infection. So there are these diseases that plague humans primarily now in kind of underdeveloped countries where you have poor sanitation. And I was intrigued by how they establish chronic infections and how they maintain these infections in a balance with the human body, where the human body doesn't try to get rid of the organism. It strikes a balance to keep the human alive and then keep the organism alive too.
Kind of like a host.
So very immunologically focused. And then when I finished my PhD, I had a couple of decisions. I had an offer on the East Coast and an offer on the West Coast. And I also had a 2-year-old child.
Oh, good.
Yes. And then I received an offer to go into an academic program that was primarily teaching oriented, where they said they need a change leader and a thought leadership. They needed someone to help bring the rest of the organization, especially academic, professionals, into a mindset of scholarly academia, more than just teaching, but understanding the fact that you could teach and you could educate, tomorrow's, in this case, mainly physicians, and then also be very active in the scholarly world in terms of research. And so I took on a role to take that on. Also loved every bit of that. Opened up a non-for-profit also within that institution, directed over 100 different research programs, both undergraduate and graduate, ran the non-for-profit, and during that time really took a deeper dive into the original research program I had taken on, and realized this is more than just establishing chronic infections. This is affecting the lives of people in third world countries. Yes, women and children, economic cycles, miring these communities in poverty in a way that makes it difficult for them to ever escape that. And started leading research programs in Bangladesh, and from there really developed a passion about preventing and treating chronic diseases. And during that time, I was tenured. I also then took a sabbatical position in a company that I'd been consulting. I had been consulting 5 or 6 different companies in business practices and science and grant funding, and just really, really liked this company that was really a community unicorn, a community darling. It was called Aldevron and still is Aldevron. Really had grown to have a reputation as the world's best CDMO, manufacturing plasmid DNA for clinical trials,
Mm-hmm.
and loved being there. So I ended up accepting to be bought out of my sabbatical position, left my tenured university position, and went to Aldevron. And went through a couple of different positions there, ended up serving on the executive team, learned so much, really from some great leaders there. And it was from there that I just began to cast some ideas about what I knew was kind of lacking in the pharma industry and the demands and the needs that people had versus what other companies could supply. And that word got around, and that led to the offer I got at IPA.
Wow, we'll talk about that in a moment. I tell you what, I don't know what happens with a person like yourself, where you wake up of a morning and go, this is what I want to do. It's such an incredible set of topics, and you've done so much. Do you find it difficult to rest at night? You seem to have a very busy mind. Is that a challenge for you sometimes?
It is. Now, so that's interesting. I actually have an official diagnosis of insomnia, but I think what it really is that, yeah, like my brain just keeps going.
Keeps going. Me too.
People imagine that you lie there and you're just exhausted, right? And you're just, oh, I wish I could sleep. I wish I could sleep. But that's not what happens to me. I sleep 2 hours and I wake up and I'm
good.
like, I'm ready to go.
You feel refreshed again. Thank you so very much for the feedback. Now let's take a look at IPA. What is IPA? Explain it to us.
Yeah, so Immunoprecise Antibodies is the full name of the company. It is a multinational company that exists really to solve the major demands that exist in pharmaceutical and biotech companies with regard to building novel therapeutics. So antibodies against really any type of target, any type of disease. We pride ourselves in being very data-driven and very innovative with our technologies. And in addition to that, being able to apply some of the world's most unique AI-driven applications in order to do things better and faster, but without sacrificing not only efficiency, but actually specificity and quality of the work that is done. So we can take, in some cases, for instance, work that would take years and turn that back within weeks at a fraction of the price with the same quality or better precision. And another part of what we really focused on doing with regard to IPA was meeting the major demand of not only the time and the cost associated with drug development, but also the risk, the fact that people had to use 30 or more companies to build a drug from the time they had an idea to the time they went to the clinic. And we solved that problem not only by investing heavily in R&D and building some of the world's premier platforms in antibody discovery, but also by acquiring. We've made 3 acquisitions since 2017. When we felt we couldn't build it better than anyone else, we went and bought who we thought did it better than anyone else.
Of course.
So that end-to-end chain really of being able to build novel therapeutics with a very high success rate and high probability of success in the clinic is really what we're known for.
What an incredible field. Now tell us about, you've touched on, I guess, the core mission, but nail it for us if you could explain what you hope to achieve in, I guess, the near and long-term future.
That's such a pertinent question today because things are changing so much.
Oh, yeah.
It's just been between like technology and how it's integrated in drug discovery with AI, which I think, I believe has gotten a lot of promotion over the last couple of years, but hasn't really panned out the way that people had hoped, versus the way that we've been applying it and the way that we see it making tangible differences and we see it out competing. And so our perception of what's possible, in the near future, I think has evolved even just over the last 6 months, seeing the products that come out and seeing the quality of the work that comes out of our AI and our algorithms. So when we really look forward at kind of the small, the short term to the medium term and where we see ourselves going, we're getting more and more digital. We're getting some of the things that we would normally accept hundreds of thousands of dollars from clients to do in the laboratory for them to turn around a custom product. We can now offer them software so that they can do the majority of those things literally from their computer at home.
That's crazy.
And it is crazy. It's terribly unprecedented, and it is absolutely cheaper and faster for them. But because we have unique ability to make these offerings for them, we also see extreme client retention and an expansion of our client base beyond just pharmaceutical companies, of which we serve the top 19 out of 20 largest companies right now, but also biotech, technology companies, other AI companies, everyone that wants to make that push and expansion into drug discovery.
Yeah, that's incredible. Thank you again for sharing. Now, I know that you have some offices around the world. Tell us a little bit about where IPA is kind of working from, head offices, labs, and all those sorts of things.
Yeah, so our central lab that was the original IPA, the original immunoprecise antibodies, is in Victoria, British Columbia. So it's on Vancouver Island. It was founded, I think, close to 30 years ago by a wonderful couple who was really looking to create biotechnology jobs in British Columbia, which wasn't that common. And they did a fantastic job really focusing on creating jobs, focusing on innovation. They developed some stellar technologies that went on to be sold. Really, quite frankly, I think for pennies on the dollar, many of those are staples in every lab today. But again, that wasn't their focus. They created a few technologies that have now gone on to be premier technologies today in the industry. And so that was our first location, Victoria, and it is still our primary revenue generator, our flagship for our most well-known acclaimed technology, which is called our B-cell select technology. And that's kind of where it all originated. From there, we made an acquisition in Utrecht in the Netherlands that did the downstream manufacturing for our primary technology, the B-cell work that we were doing. After that, we thought, all right, well, we're making great antibodies for people. We're able to produce them. Now, how do we develop them? Just make improvements on them, right? Prepare them for the clinic.
Yep.
And for that, we identified a company called ModiQuest in the Netherlands also that just had an excellent reputation for that. So we acquired ModiQuest. Now Utrecht and the Oos campus, which was ModiQuest, are now known as IPA Europe. And then about 2 years, 3 months ago, we acquired our third company called BioStrand. And BioStrand is an AI and ML driven company out of Belgium who had this crazy, almost sci-fi discovery about 15 years ago, where one of the co-founders, Dr. Dirk van Hyfte, had discovered that there were discrete and finite patterns in all biological information. So DNA, RNA, protein, sequences of information that where there were only a certain number of sequences that coded across all biological organisms, very specific information for what that molecule could do. That was not anything anyone realized existed. It was not intuitive. These were not long stretches of sequences. They were just short little pieces that needed to be in very specific places. And when they realized that existed and they realized the meter stopped running, they realized, okay, that's it. There's only a certain number of these that define what every molecule on the planet does. That's it. They left their jobs. They were both physicians. They both were extremely educated, doing very well in their careers. And they started BioStrand and they started coding. They started collecting the information and integrating that into the index of an algorithm. And that algorithm today can screen pretty much any type of information. And it can do that extremely rapidly because it's taking it through a finite set of data, and like anybody else in the world, which means they also save on energy and time and on money. And they can extract from that insights by connecting all different data types because, again, these little fingerprints or patterns are existing across any type of biological information. So that's the core of, their discovery, which provides unprecedented insights and allows people to take really any type of data and then harmonize that and use that for research, which is critical in precision medicine. And just so turns out that it's answering one of the biggest pain points in pharma today, which is what do we do with all this data that's disparate and from different pieces of equipment and different clinical trials and electronic health records. So it's amazing. And that's really where we've gotten all the analytical tools that we've built as of late. And then aside from that, we have an executive office in Fargo, North Dakota. When I left Aldevron, I was in between the Utrecht site and the Victoria site. And I took a handful of the management team with me, which was appropriate and collegial at the time. They were actually had undergone majority management change. And they were really focusing on, that plasmid DNA and messenger RNA, which ended up becoming crucial in meeting the supply demand for COVID. And so what I really brought with me was much more of the therapeutic antibody side of the space and sales and project management. So yes,
what an incredible story. They say that teamwork makes the dream work, and I tell you what, you can't have one hand clapping in a business like this. I wonder, to make breakthrough discoveries, which I'm sure you're doing across the spectrum of different conditions that you could help to identify antibodies for. Are you touching on everything, and how do you work together to make these breakthroughs? The coordination must be something else.
Yeah, that's a great question. I mean, we do really address any type of target. And any type of disease. So the targets range in complexity, and these days they are getting more and more difficult because the easy targets, we've done that, right?
Yep.
And we love those challenges, and that's another area where we're just well known for being able to take on really difficult programs and be successful. And certainly our B-cell platforms in Victoria help with that. There's no doubt about that. Just the input, the throughput, and the output are phenomenal. So we really do tackle kind of any type of problem. But one thing that is unique about us is when people come to us with a program, whether it's a partnership or it's fee-for-service work, we're very custom about how we approach it. So we really look at the whole program. We use our analytical tools to understand the target and the disease and what's really happening. And again, connecting data where we can draw from that information that others wouldn't see because of our pattern recognition. And then we design a custom plan. And so that enables us to really determine the best path forward and how to do that. And, that involves scientists at all of our different locations, and even sometimes involves management, right? When we're really taking a look at the best path forward to set our clients or our partners up for success. And so it really is teamwork. It really is bringing a lot of different perspectives. It also comes with the collective, 70 years of experience in this space. Just really understanding what platforms and technologies work for what and where we've seen success, and little tricks of the trade in terms of how we've been successful in the past.
I look at the global nature of the IPA organization. I think to myself, is there a sort of an overarching global regulatory obligation that's, cross C to C that you have to abide to? How does that all work?
Yeah, no, that's a great question. I mean, this is actually one area where we're really lucky because we do this full end-to-end, but we stop when it comes to really serving parts of the industry that hit the really tight regulatory components for use. Yeah. Yep. So every site has provincial or state or whatever regulations on the use of genetically modified organisms, how to dispose of material, how to handle bloodborne pathogens, how to dispose of chemical waste. We all have those and different protocols to stay up to speed on to keep our employees safe. But when it comes to the big hurdles, right, the ones that, you need continual audits on, you need traceability and all of those sorts of things, we stop just short of there. And we have people tech transfer or turn things over to another group to take that. Yeah, lower risk too.
I love it. Yeah, that's the way to fly. I think about AI technology, and I think about common everyday usage. Then I think about the proprietary nature. It must be proprietary nature AI to develop this sort of stream, if you like, of products and service solutions to your ideal clients. How does that get developed? And do you have your own thousands of servers? It's an amazing topic.
It is actually. So that's really interesting because that has taken some harmonization on our part to really standardize how do people handle the data? How do people, actually annotate that data? How do they store that data? How do we share that data? How do we really make sure that all the information from the client is protected? And it's not just about the data there. We're so used to already protecting ideas and information coming from clients because, obviously we never share and we never reuse anything that comes from someone that's their idea. But on the whole, data level, that is a whole nother level. And we've really had to up our game and get very serious about cybersecurity. We were really good at it already. We inherited just some amazing people with that BioStrand acquisition. but yeah, we absolutely, needed to be safety forward, with regard to dealing with all of that. And then when it comes to our algorithm that really drives, I would say, this kind of differentiation that we have with our side BioStrand, we have patents all over the place. We have the most, intense, thick patent wall around that to protect, our proprietary information within that algorithm. And then we also have, just these really interesting switches where it's like research is being done and analytics are being run. And actually, you're not actually tapping into the algorithm most of the time. There's a very short window where it opens and then it closes. So we have, great safety protocols to protect that information for the company.
I've been looking through large swaths of data on your website, and I came across multi-omics modeling. Could you share a little bit about what that means?
Absolutely. So, I mean, this kind of goes back to a little bit of the pain points in the industry. When we talk about data and data being able to be harmonized, one of the things that started becoming clear like 10 years ago was everybody had all these different types of data. They had information coming out of the clinical trial. They had data just being, pushed off of equipment in the laboratory. They had handwritten notes, all of this information that really couldn't be used and studied together, which is so critical if we want to make safer drugs and if we want to make more specific drugs. Oftentimes we need details about, the exact mutations in a patient, the drugs they've taken that didn't work, understanding the structure of a molecule, really pulling as much information together to be more informed the next time we build a drug. And what we found is that oftentimes that information is housed in DNA or RNA or in protein sequences or structures. And that's part of what we're referring to when we talk about this multi-omic data. It's these different types of sequence information that can be harnessed at massive levels, tremendous amounts of information, in order to inform us about how to build a better drug. But in traditional software settings, and this is still something that people kind of scratch their head on today, like realizing that we can really do this. Traditional settings, you can't analyze all those different types of omic data in kind of, really, one system.
Yeah.
And that's something that we're able to do with Lens AI. And so it's able to pull out the patterns from across all of these and find the commonalities and the themes that we wouldn't see with the human eye or with other algorithms. And so that's hugely important in drug discovery. And probably, the easiest example for people to understand is in the personalized medicine or the precision medicine space. We have talked now since like 2000, since about people being able to get like their entire genome sequenced and understand like what all their genes are like at the genetic level, and maybe even a little bit about what they encode for, which we're much better at today.
Yep.
And, what might have gone wrong with a particular disease, which could inform us on how to treat that. But we've never had software that could process that information fast enough. And now we're at a point where we can do an entire genome of a human for a couple $100 overnight.
Oh, that's amazing.
Unprecedented. Yeah.
Talk about technological quickening.
It really is. But then the challenge became, well, you have the information, how are you going to analyze it? You might have the genome information, you might know which genes are being expressed, and you might even know the molecule and the shape of it that's gone awry to cause this disease. But as far as we know, we have the only software that can look at all of that and can analyze that genome so rapidly to bring back information in time to align the patient with a drug and in time to save a life. And that's where we think really the future of this goes, beyond drug discovery, beyond drug development, but into the clinic, helping with patient enrollment, helping to understand which drugs are the best fit, et cetera.
Yeah, look, I tell you what, and that is the core mission, is to help save lives and, avoid exposure to ailments that can be crippling to one's life and life experience. Thank you very much for sharing. Now, I know that you've had certainly some clinical successes. You've also had a Nobel Prize. Tell us a little bit about those two things.
Yeah, so for clinical successes, that's been an interesting ride. We have so many clients and partners that are incredibly successful at what they do. And it's, oftentimes they're coming to us with a sense of what they want to do, sometimes a really good idea of what they want to do. And We don't always know when the drugs we make for them go on to be successful, but sometimes we do. And we have developed drugs against a multitude of different diseases. The most recent one that we developed that it has having some great success at this moment in the clinic is one that is directed against what we call epoxic solid tumors. And what I really like about that example, I mean, all of them, I have a heart for all of them because they're fun and we know the people who are building them. But this one I love because it was done on one of our B cell platforms, and that actually takes the initial sample from a rabbit. And, we don't need to harm the rabbit to do this. We just take a blood draw and we use that sample to harvest what we call a B cell, which is where the antibodies originate. And we're able to get into that B cell and find the genetic sequence to encode for that antibody and to collect just a tremendous amount of information in a 1-week period. And it was that platform that is so uncommonly used to make a clinical product or a commercial product that also has so much potential compared to a traditional platform that generated this therapeutic. There's another one right behind it that has gone on to be so successful so far. And so, that's just, it's something that we love. We don't back away from any disease, but we certainly get a lot of oncology, inflammation, infectious disease. And then as far as Nobel Prize, we actually ourselves haven't had a Nobel Prize. We have come alongside to support and cheer on other groups that have had Nobel Prizes that have really to support the industry. But if I could use that just as a plug to say, hey, when this all breaks loose and people realize how powerful lens AI is, I think Dr. Dirk van Hyfte should get a Nobel Prize.
Oh, absolutely.
Yeah, pinpointing and finding these natural patterns that exist that no one saw before, and having the patience for a decade to uncover them and code them and log them and then get to that aha moment of, well, this is pretty serious.
That is no mean feat, is it?
Not at all.
That's what is that? That's going to have global impact if it, over time.
I honestly think so. And I think far beyond therapeutics, we had to focus on something initially. And IPA was good at therapeutics, and we saw this huge potential in this algorithm. But honestly, anything biologic can be touched. So I mean, vaccines, agriculture, and we've already done some work for the agricultural community. Anything biologic in nature, it can take, the patterns from and then connect them and then glean information that would otherwise be impossible. So we're super excited about the potential of this platform.
I'm sitting here getting all excited thinking about the future for humanity. It's just incredible work that you're doing. Now, I know that on, I guess the governance side, you have a team of very, very highly skilled professionals. Tell us a little bit about governance and the team behind IPA directly.
Yeah, absolutely. Yes, we do. We have individuals from all different backgrounds and all different experiences, bringing together a global team that has come from all different types of companies. And so, most definitely, obviously, we have a tremendous team of scientists. We also have a tremendous team, including at the board level, of people, from various business backgrounds, various finance backgrounds. And interestingly, too, I think coming from a tremendous number of cultures, and not just because we span a lot of different countries, but because also each of our sites is also tremendously diverse, and some of them representing double digits of countries that are present at that location. And so it's really that, diversity of thought, that diversity of experience, in many cases, multiple individuals at a location with, management and even executive experience. And, working across not only different cultures, but across different disciplines, which is so important, right, in our space. And so, it's really, I think, developing and cultivating and building upon a pre-existing respect for different thoughts, different views, different perspectives, but all doing that also with an existing framework of, really what's right, really how to align ourselves in an appropriate long-term way to be respected and to be a long-term player in an industry that we all, I think, have a tremendously good handle on and background on, but also respect for.
Absolutely great feedback. Now, I just wonder from a personal perspective, Jennifer, how does it make you feel knowing that you are making such an impact in the world? How does it make you feel?
Mostly, I would say, really, to be honest, proud of my team and proud of it's really the other people out there, really, like where that rubber meets the road, the day-to-day application of the technologies and the groups coming to us as well that have that vision too for who really needs those treatments, who's really struggling and who's really suffering. And when I look internal to IPA, I think the thing that makes me the most proud is the commitment to innovation. Like sometimes the needs of the industry move faster than the technologies. And we have to innovate on the spot. We have come up with new technologies right there on the fly. We've worked with pharma and biotech companies to do things that we've never done, and we're pretty sure other people haven't done. But it's really needed in order to tackle something that's arisen. Maybe it's an infectious disease. Maybe it's a company coming to us that's kind of hot on the tracks of something that no one else has uncovered before. And being nimble, being agile, being courageous, not being afraid of change, not being afraid to step out there and try something new, not being afraid of failure. I think that's a huge part of what we have staked our reputation on and why our reputation has grown to be as good as it is.
Well, it's a real credit to you as the CEO and the team around you locally and globally, obviously. Now, I know that you do have a lot of events and online webinars and that sort of jazz. Tell me what people are likely to, I guess, find, where they're likely to find you, and how they can get involved.
Yeah, absolutely. Yes, we are constantly out there. So we do a lot of conferences. Those might be technical conferences, scientific conferences, technology conferences, and investor conferences. We do a lot of events where there's just one-on-one opportunities for meeting with people. And yeah, that's how we enjoy getting the word out there and what people can expect to see. You can expect to have, quality one-on-one team with members or time with members of management. You can expect to find live demonstrations of what our in silico algorithms are capable of doing. In addition to that, as we go through this process of going to conferences, of making ourselves available, people can always attend those. They can always sign up for those. But you can also get more information, of course, from our website. It's ipatherapeutics.com, or you can go to biostrand.ai. We also try to, whenever possible, upload presentations, public speaking events, demonstrations, just think different ways we might be involved in the community as well. We're involved with a lot of our partners and supporting them through, various types of events, public events, and supporting different causes. So whenever possible, we put that information on our website as well, just so people can see what we're doing, see what we're up to, get involved, and better understand our mission.
So, with all that being said, I wonder about the listing. Do you have a ticker, and where can people find if they're interested in that side of things?
Yeah, absolutely. So we are listed on the NASDAQ Global Market, and our ticker is IPA.
That's an easy one to remember, isn't it?
Yes. I'm sure there's a lot of your companies that are jealous.
This has been a wonderful call, Jennifer. And anybody who's on the call with us today, you want to learn more, we're certainly going to be making sure that all the links are available below this post. No matter where you see it, you're going to find those links back to Jennifer, her wonderful team at IPA. And with all that being said, Jennifer, thank you so very much for joining me on the show today.
Thank you so much for having me.